Hello, everyone. Welcome to today's EEB seminar. My name is Ashlyn Ford, and I will be your host. I'm a third-year PhD student in Peggy Fong's lab, and just quick ground rules here. If you have any questions, please save them till after the end of the seminar. You can ask them via the Q&A box, or you can raise your hand and we will activate the microphone for you. With that being said, let me introduce today's speaker. Dr. Esther Ngumbi works as an assistant professor in entomology and African American department at the University of Illinois at Urbana. And her research involves basic and applied aspects of using microbial inoculants to promote growth and enhance tolerance to drought stress in multiple crops. She has received 3 U.S. Patents for her research, and she also was inducted into the National Academy of Inventors. The technology developed from these patents will impact millions of farmers and result in commercial products that will generate revenue worth millions of dollars. Besides research, she is the founder of organizations that empower farmers and youth in Kenya, and she served as a mentor for entities like the Clinton Global University Initiative and President Obama's Youth Leadership Program. And with that being said, she will be talking with us today about untangling chemical-mediated interactions between beneficial soil, microbes, plants, and insects. Please give a warm welcome to Dr. Esther Ngumbi.
Okay, thank you so much, Ashley, and thank you everyone that's spared some time to join me today and to listen to my talk that is going to be on untangling chemical-mediated interactions between beneficial soil microbes, plants, and insects. And as Ashlyn said, yes, I'm an assistant professor here at the University of Illinois at Urbana-Champaign. So without further ado, I would like to first of all thank and acknowledge our mentors, our students, and collaborators. Without my mentors, I wouldn't be who I am. They have truly held my hands throughout my career, beginning from Kenya. I have a long-standing, over 20 years mentor, Dr. Baldwin Toto at Auburn University, where I got my PhD, Professor Henry Fadamiro and Professor Klepper. And then at Georgia Tech, where I did my postdoc and Mark Hay. And then here at the University of Illinois, May Berenbaum, Larry Hanks, and Andy Suarez are close mentors that continue to guide me. And also thankful to my collaborators who have had an amazing time thinking through the science that I do and working together to generate our results. And I wouldn't also be a stronger scientist without students who do a lot. And as an assistant professor, I still have a small team. Erin Duddy is my undergraduate student, and I'll be recruiting more graduate students very soon. So this is my beginning team, and we do have a lot of fun. And I also want to thank Dr. Zapata and EEB graduate students who invited me for extending this invitation, and Sheena, who has been also key in ensuring that all the technicalities before this talk happen. So thank you to all. So I always say, if at the end of the talk you don't take anything, I would like you just to think of our world as a volatile world. I tend to think of it as when you think of plants, they can't walk, they can't communicate. However, we know that chemical signals, or rather volatiles, are their language. I tend to think of them as 911 calls. So every other second, minute, or hour, a 911 call is going on. A plant is calling for help. from our natural enemies, or it's calling for help, recruiting our beneficial soil microbes, and it's also warning other neighbors, plant neighbors, of impending attacks. So it's a very busy and busy chemical signal world in the plant, insect, beneficial soil microbe realm. And the passion to truly understand how these volatile signals mediate conversations among beneficial soil microbes, plants, and insects has truly allowed me to go through a global journey, beginning in Kenya, where I worked at how volatiles do serve to monitor, as we can use them to monitor the establishment of natural enemies of insect pests. Then my love for chemical signals and chemical-mediated interactions took me to Israel, where I spent actually 2 years looking at how also chemistry-mediated interactions happen in stored product insects and how they use the chemical signals for defense. Then to Idaho, where I looked at how actually chemistry also serves to recruit plant disease vectors. And then Sweet Home, Alabama, where I did my PhD, and I looked at just how chemical signals mediate plant defense and natural enemies recruitment, as well as recruitment of beneficial soil microbes. And then finally here in Illinois, where I'm also continuing my research on looking at microbes and untangling how chemistry is mediating the conversations among plants and insects. So as I said, when you think about plants in nature, they don't walk, they cannot move, and yet they must grow and produce. And they are a part of very complex communities that we now know include below the ground, where there's a lot of associations, especially with the beneficial microorganisms, including rhizobia, mycorrhizal fungi, plant growth promoting rhizobacteria, that then also above ground, plants interact with both beneficial insects on one hand, and on the other hand, insect herbivores, which come in many different styles. We have the chewing insects, we have the phloem sucking insects, and many, many more. And so to truly understand volatile roles from the plant's perspectives, we need to basically work with organisms that include insects, beneficial soil microbes, that have actually paid attention to these chemical signals that a plant emits at any given second or an hour. And these associations have been evolving for millions of years. So they've been fine-tuned by nature, and they are adapting and changing with every day. And so as a scientist, I keep on always finding myself just running and really just being behind of what is happening nature-wise. And that's what gives us joy as scientists, to know that we are working on things that are not consistent and changing, they're changing and very dynamic. And we know now that indeed these interacting members of plant communities can and do actually trigger the emission of volatiles. So 20 years of research that has continued to be amazing and fascinating has truly just elucidated the fact that anytime a plant is attacked by pathogens, insects, viruses, and also below ground, all these interactions do actually trigger the emission of these volatile organic compounds, these chemical signals that then start the lines of communication. They open the line of communications among these interacting members. And we know that these emitted volatiles that actually do mediate a lot of the tritrophic interactions. And because they do play a bigger part, they can shape the ecological functionings of ecosystems. We know that, yeah, volatile signals do play roles in mediating plant herbivore, plant to plant, microbes to plant herbivore, natural enemies. And you can keep on building this multi-trophic interactions. And the bottom line is that indeed, yeah, chemical compounds do mediate these conversations. Over 20 years of dedicated research so far has allowed us to identify over 1,700 volatile organic compounds. And I think as technology continues to improve, we are truly going to unravel many, many more organic volatile compounds that play roles. And when you think about it, these messages, as I say, these are 911 calls. The messages that are contained in these volatiles are very specific. We know that. And they're highly reliable and truly do constitute of honest signals. And we know research has clearly shown that these released volatile signals can reveal details about, first of all, the plant type. So different plants will emit a signature volatile compounds. They can reveal the type of herbivore that is interacting with the plants, whether it's a chewing caterpillar or a phloem-sucking aphids, thrips. And even within the herbivore species, you can also tell which herbivores. Is it the fall armyworm? Is it the corn earworm? And also, it can reveal at what stage this attacking herbivore species are. Is it first insta caterpillar? Is it second insta caterpillar? Is it third insta? Or is it even an ovipositing moth that is trying to oviposit on plants? So the chemical signatures allow us to truly identify the herbivore species. And moreover, we also know that these volatile signals can also help us identify the signatures, can help us know what type of stress the plant is facing. Is it biotic, which is herbivory, or is it abiotic, drought stress, flood stress, and many other stresses that the plant encounters every day of its life cycle? And we know that for sure the quality and quantity of these volatiles can be influenced by many, many factors. It can be influenced, as I said, by the herbivore species, by the plant disease that's affecting the plant. The microbe strain can also influence the quality and quantity. The plant, whether it's a tomato plant, whether it's a corn plant, soybean plant, and as well as the abiotic factors, temperature, humidity, and pollution, soil factors. All of these factors can change the chemical signature of a plant. And once that change, it can further then influence the plant and insect interactions that are mediated by these signals. And so because everything, it's all about the volatiles, the volatile world, it's important that we understand both the variations in the total blend, the individual compounds, as well as the categories of the different compounds, whether it's green leaf volatiles, the monoterpenes, the sesquiterpenes. And so it's important for us to dissect this volatile blend, and so that we can be able to understand the biological and ecological functions of this chemistry. So my lab here at the University of Illinois seeks to understand the signals that mediate these interactions among beneficial soil microbes, plants, and insects. So we do use a comparative approach using model trophic systems and multiple chemical ecology tools. So, and since I said we must first of all collect that plant chemistry, and so using glass collection chambers and other techniques, including solid phase microextraction, we can first of all just collect this chemical chemistry and plant chemistry and use adsorbents to collect. And then once we collect, we do use analytical tools that are available to us, gas chromatography that's coupled with a mass spectrophotometry. And then finally, because in the end, we want to make sure that our science has applications at the farmer level. We couple all these analytical and chemistry collection techniques with actual behavior studies to truly understand what compounds, what blend within the total blend is important for the insects that we are working with. And so my lab here at Illinois, we are working with model systems, and we have two so far. On one hand, we are working with mycorrhizal, fungi, plant, and insects. So we do use both the commercial available inoculants as well as just individual strains. Our crop so far is tomato, and then our insect is Manduca sexta, and the tomato hornworm. She's pretty. And for those of you that don't love insects, I mean, I think insects are just beautiful. They're beautiful even to work with, and it's amazing, and it's fascinating the interactions they trigger and what happens. And so the other model system that we use is bacilli PGPR. This is a plant growth promoting rhizobacteria as the beneficial soil microbe. And my plant of interest is maize or corn. And I mean, you cannot stay in Illinois without studying corn. You would just not. I mean, we grow a lot of corn here, so it was just natural to settle down on maize and then use the fall armyworm as the insect. So I would like to talk about my first project, which is I'm looking at unraveling chemical-mediated interactions between mycorrhizal fungi, plants, and insects. And on the left are my collaborators, Dr. Marginot, who is a soil scientist, and Dr. Hind, who is a microbe scientist. And then on the right are students that I've worked with, Erin Daddy and Easy. They came through as it was an NSF-funded research undergraduate immersions, and it was truly an amazing time to work with them. Easy now is at Penn State, and Erin continued to work with me, and she's just amazing. So when we think about arbuscular mycorrhizal fungi, which are there's a beneficial soil microbes, is the most common and functionally important symbiont of terrestrial plants. We know that over 80% of plants do have this association with mycorrhizal fungi in the soil. And when you think about this mutualism, the host plants do provide the shelter and carbon and a mode of reproduction for the fungi. And in turn, fungi provides as many benefits as we have been able to identify, including promoting plant growth and acquisition of soil nutrients. And it's indeed a very well-studied system, I would like to say so. But apart from the nutritional benefits that come about when plants form this association with mycorrhizal fungi, we know that indeed also there are other many benefits, including actually alterations of the chemical composition of the host plant, and in a way that the plant can be able to attract more natural enemies or be able to fight better the invading insect pests. And this is results from many studies. I said it's been very well studied. This is a system that has truly been studied. But one of the really gaps that is evident once you look at the literature is to realize that many of these conferred benefits, including alterations of plant chemistry, are indeed very variable, and they are dependent on many both independent as well as interacting factors, including the plant type, the genotype, or the variety of cultivar, the abiotic or biotic challenge that a plant is facing, the AMF strain that is used, as well as many environmental factors, temperature, water content, and importantly, soil fertility, we know is a big factor that can shape the outcome of mycorrhizal plant-insect interactions through altering the plant chemistry. So the research goal that my lab is seeking is to understand how soil fertility influences this mutualism and further then affects the induced volatile emissions. As I said, my model crop is tomato. And so the question here we're asking if the outcomes of colonization by mycorrhizal fungi in terms of the changing plant chemistry is modulated by fundamental agroecosystem characteristics such as soil fertility. And when we talk soil fertility, soil phosphorus. So our working hypothesis for this research question is that under conditions of nutrient limitation, we hypothesize that mycorrhizal fungi mutualism will indeed enhance the defenses and further impact volatile signaling with impacts on the plant and insect interactions that then further follow up. And for this research, our predictions were that indeed, once colonization happens, we will see an increase in the emission of tomato volatiles, and that soil fertility, especially when the plants have low soil fertility levels, we will see more cooperation, more very stronger mutualism between the plants and mycorrhizal fungi, and see that in some of the non-nutritional benefits, which is changing of plant chemistry. So our approach for here, we use two genotypes. We have Mike, which is the wild type, which fully colonizes very well with mycorrhizal fungi. And then we have the reduced mycorrhizal mutant that does not form associations with mycorrhizal fungi. So with these two genotypes, we are able to truly see the benefits of mycorrhizal colonization. And as I said, our insect is our beautiful tomato hornworm. And so our approach is to grow the tomatoes under greenhouse conditions. And then 6 weeks later, we bring them back to our labs and infest the plants with Manduca sexta. And then we collect the volatiles using solid phase microextraction and further then analyze and identify the chemistry. So for this study, when we looked at the overall blend of volatiles, we indeed were able to show that under low soil fertility conditions, the mycorrhizal wild type, first of all, is colonizing. We see the benefits of mutualisms, but also we see actually the change in chemistry, and this is the total blend, and you can see the UD is damaged, and this is low P is low phosphorus and high P, and Mike, which is the mycorrhizal wild type, as compared to the mutant that doesn't form mycorrhizal mutualisms. So we were able to see differences in chemistry, and more so to certain that our hypothesis was correct. But as I said, with the chemistry, we look at both the total blend and we look at the groups of the different compounds, monoterpenes versus sesquiterpenes. And because we know that different categories may be recruiting insects at a higher level. So for example, we know that because of herbivory, there is a lot of increase, especially in sesquiterpenes. These are the the so-called induced volatiles. And they're called induced because they are only produced once the plant is under herbivore attack. So we see indeed that with the monoterpenes, we don't see a big difference between the mycorrhizal wild type and the mutant. But when we look at the sesquiterpenes, we indeed see differences in plant chemistry with low P and the mutualism forming genotype increasing its plant chemistry. And we see that trend when we also now look at the individual compounds. And here I show trans-beta-ocimene is one of the compounds in the literature that's actually been shown to be, once it's emitted, it can attract other natural enemies that can then attack the caterpillars and reduce the insect density as well as the insect factor in the plants. And we also see that in caryophyllene, which is an induced volatile. So indeed, we were able to see differences in volatile compounds, and that soil fertility is indeed impacting plant chemistry. But we also were able to see other benefits that are associated with mycorrhizal fungi colonization, such as improved growth. So in summary, for this project, we were able to show that indeed soil fertility influences mycorrhization, and consequently shapes the volatile emissions in tomato plants. And we further predict that these changes that we have documented will influence herbivore location of host plants and oviposition preference as well. So these are the next plant studies, is we are going to further grow the plants and look at oviposition preference by the tomato hormone. And so we are further working on that, and Erin is the student in charge. Then moving on the other project that we are working on, it relates with the mycorrhizal fungi model system. So here we are further wanting to understand also the influence of plant variety on mycorrhization, as well as volatile emissions in tomato plants upon herbivory. And my collaborator, Dr. Claire Whiskey, and Erin Duddy, again, is the student. And so for this research, we know that, as I've shared, that there's so many benefits that happen when plants form these associations, including forming association with mycorrhizal fungi. And we know that, yes, plant chemistry is altered, although we know that the results are very variable. We also know that there are many factors that can influence volatile emissions, including the plant variety, cultivar, or genotype. And when you think about crop breeding, every year we are the selective breeding, and we are breeding crops that we're selecting for fruit size and many other characteristics. And it's possible that as we alter crops to satisfy our demands as consumers, we are not considering plant defense mechanisms. So we want to understand how crop breeding through alterations that happen, if that also influences plant induced defenses and plant chemistry, and then how that cascades to influence our plant and insect interactions. So for this system, as I said, we're asking how does plant variety and mycorrhization influence induced volatile emissions in tomato? And we're using field soil. Part of my lab is truly, while I love basic science, I would like to see that science translating into solving the real challenges that farmers are facing. And if you followed a lot of the time, most of the time, a lot of the greenhouse studies do not translate into the field. And so we are trying to bring in as much of the field components that we can bring into the greenhouse to ensure that our results are almost, at least they have a meaning in the field. So that's why we're using field soil from the Champaign County. And for the varieties, we're using 2 heirlooms and 2 hybrids. And these are varieties that actually most of them are locally grown here in Illinois and in the Midwest. And our insect is Manduka. And as I said, we collect our volatiles using solid phase micro extraction. So once again, I've said we have 4 plant varieties, 2 heirlooms, which are the Amish Pest and Cherokee Purple, and 2 hybrids, the Valley Girl and Mountain Fresh. So these ones are bred under different conditions. And so our approach is we grow the tomato seeds, and then 3 weeks later, we transplant. And it's also at this time that we inoculate them with mycorrhizal fungi. And you can see Erin dipping the transplants in a mycorrhizal solution. And then we grow the planted tomatoes under greenhouse conditions for 4 weeks. And 4 weeks later, we bring them to the lab and bring in the insects now and further collect the volatiles and do other amazing stuff. And so moving on to the results, once again, we've said that there are many benefits that come with inoculations of plants with beneficial soil microbes. So we were able actually to see the differences. And so here you can see, so I've AP is Amish pest and AP with no AMF. And you can see that indeed we are seeing differences when we have the mycorrhizal fungi inoculated plants. We see differences both in shoot as well as root weights. But we don't see that much with the hybrids. The differences were more glaring when we had the heirloom varieties. And we also see differences in the root weight as well. You can see that indeed inoculating plants with mycorrhizal fungi does improve roots. And because the root structure and the roots of the plants are improved, then the plant also grows better. And so we also expect that we will see changes in plant chemistry. And so these are just also scanned images of the roots. So you can see that it's truly a dynamic difference between plants that are not treated with mycorrhizal fungi and the ones that are treated. So when we now looked at the plant chemistry, it was surprising, first of all, because the mycorrhizal treated plants were so big. I mean, they were just like really, truly a difference in growth, but they did not produce a lot of volatiles. So it was clear that, first of all, yes, plant variety does change the chemistry, and different plant varieties are emitting different quantities of the volatile organic compounds. However, the mycorrhizal treated plants did not emit higher blends of volatiles compared, and when I mean higher in quantity-wise. So as I said, when we are dissecting the blend, we are looking at the quantity, the total quantity, as well as the different categories of compounds, as well as also the individual compounds. So indeed, we saw that AMF inoculated plants produced lower amounts of volatiles. And this was also evident not only with the total blend, but also when we looked at individual compounds, including beta-phelandrin is one of the major compounds in the blend of tomato. We see a differential emission in terms of the quantity. And so in summary, indeed, while we saw the benefits of inoculating plants with mycorrhizal fungi, it is true that there could be trade-offs involved between investing in growth versus investing in plant defense. And so this really got us very interested, and we're really like fired off to now understand what are the underlying mechanisms? Why did we see these huge differences in growth, yet differences in chemistry were not that clear, or were not that as exciting? So we want to do this study again and do it in the field, and to further understand what are the implications, first of all, of the blend, the mechanisms behind these changes, and more so, what are these differences between the hybrid and the heirloom? Why are we seeing such a drastic difference between these different varieties? And so, and further also understand what does these changes in the plant chemistry mean to ovipositing herbivores? And further on, also understand there are also consequences for growth and performance of Manduca sexta. Between if we now are seeing that the colonized plants are growing better, does it mean that the insects would grow better? And then, or are there other defenses that we are at play? Remember, there are so many defenses, could be also direct. We have secondary plant secondary metabolites that can influence and impact the growth and performance. So all this amazing stuff we are excited about finding out, and also are doing this work in the field. So moving on to another project. So it's all about microbes in my world. I love this amazing tiny microbes that have big impact when you think about improving plant growth. So the other project that we are working on at my lab is also further understanding chemical-mediated interactions between plants. And this time we are moving from mycorrhizal fungi and using plant growth promoting rhizobacteria. And these are bacillus, microbes. And my collaborator, Dr. Angela Kent, who is a professor here as well, a microbial ecologist, and Sierra Raglin is our PhD student, quite an amazing student. She was microbes, and then finally I was able to introduce her into the world of insects, and she is doing well. So you can come over to the insect world. And so, as I said here, we're working with Bacilli PGPR. Our maize, our model crop is maize, and our insects are Spodoptera frugipada, also commonly known as the fall armyworm. And the reason that I chose fall armyworm, because it's not only a voracious pest that impacts crops here, but also across Africa. I come from Kenya, and I'm also happy to pursue on projects that can translate not only in North America, but across different continents. And fall armyworm has, if you follow the news, truly impacted a lot of farmers across Africa, and then moved to Asia. So what do we know about bacilli plant growth promoting rhizobacteria? These are also rhizosphere, meaning that they are in the roots and near the root interface, and they colonize roots and cause many beneficial effects on plants. And most of the bacterial inoculants consist of plant growth promoting rhizobacteria, and they are bacillus. Mostly bacillus. And so these are just images, are microscopic images of roots that are colonized already by PGPR. And so what we know, once again, with a lot of literature that has been done, is that associations of plant with plant growth promoting rhizobacteria, so I will continue on referring as PGPR, like mycorrhizal fungi, can promote crop growth. It promotes both below ground root growth as well as above ground root growth, and in multiple crops, from soybean to tomatoes to Chinese cabbage to corn and many other. One of the benefits of using PGPR is it works in multiple crop systems. And we also know that association of plants with PGPR can improve the ability of plants to tolerate environmental stresses as well from drought. And these are some of the work that I did at Auburn, just showing how inoculation of plants with PGPR, both in cotton and corn, does actually help. And after 5 days, when plants are treated and not watered, the PGPR treated plants are still surviving and making it. And when you look at the control plants, they are literally dead. And this is also a study that was done way 2001 that showed that also inoculation of plants with these cucumber plants helped them to tolerate insect herbivory and this striped cucumber beetles. And the mechanism actually is that PGPR increases the cucurbitacin levels in the plants, and as a result, cucurbitacin is a secondary metabolite, and as a result, then does affect the insects. So we also know that, and this is part of the work that was part of my research, my PhD research, I was able to show that indeed association of plants with PGPR does alter plant chemistry. And this one was work done, as I said, at Auburn, where it was very evident that untreated plants do not produce the same blend of compounds that PGPR treated plants. And further, we were able to see clearly that association of plants with PGPR does actually influence the growth and development of caterpillars. And caterpillars feeding on PGPR plants performed poorly, did not grow, and we see that both at the larval stage as well as the pupal stage. And the work actually by Kate and other colleagues at Auburn truly showed that indeed once you treat the plant with PGPR, the chemistry changes, the herbivores are repelled, and we have a lot of just also the natural enemies on the other ants are attracted. And it was just really a neat system. But however, a lot of the work that has these exciting results showing the truly benefits of PGPR is only work that's done with a few plant genotypes. And indeed, if we truly want to ensure that all this work, these benefits of microbes do help under real life farmers, then we should be able to work with more plant varieties. And building on my work with mycorrhizal fungi, we know that truly host plant genotype can shape the outcome of microbe interactions. But we also know that there is a growing interest by industry, and so there's potential to translate research into solutions for farmers. During my PhD, part of the work that I did opened up patents and licensing actually of the technology to companies, including Bayer, and so to make products that then can be used by farmers. And this is a growing appreciation of microbes, and so there's money to fund our research. So indeed, it's an exciting topic for us. But as I said, just like I mentioned with tomato, we know that through the process of domestication, we've altered plant genetics. And so, because we've altered plant genetics, it's possible that we also have altered plant microbe interactions, plant chemistry, and then furthering that, we can then have so many differences that then shape the outcome of plant insect interactions that are mediated by chemistry. And so these are like this summer I worked with Teosenda. I've never seen the wild ancestor of maize, and you can see the difference between that wild ancestor to the modern varieties. So through that journey of domestication, it's possible, once again, altered plant genetics and how the plant signals that attract the beneficial soil microbes, and then how that shapes into affect plant chemistry. So for this research with PGPR, alongside Angela Kent and Sierra, we are asking if the bacillus conferred protection benefits, specifically through changing plant chemistry, do vary with the plant genotype and plant microbiome. And we know that every new year, every new decade, we have new plant genotypes that have unique characteristics. So it's possible that these unique genotypes are also interacting differently with beneficial soil microbes, and then in turn we will see differences in the benefits of microbes. So with this, this project we worked on with 6 maize genotypes within 2 heterotic groups and spanning 3 decades of crop improvement. So we had the 40 era, the 60s, and the 70s. And then we had 4 microbiome treatments, Bacillus sterile, Bacillus live soil, as well as sterile soil. Then at planting, we did inoculate the maize, different maize genotypes with Bacillus. And every week, we further gave booster inoculants. And then 4 weeks later, we did bring the plants to the lab and infest them with the herbivore and then collect the chemistry. And then following that, we did a Illumina Nova sequence to understand the microbe communities that were shaping the above ground interactions and altering plant chemistry. So when we look at the results, indeed, we saw that breeding-driven manipulations, the plant genotype, do influence the maize plant chemistry. So it was clear that the different maize genotypes are clustered separately. And this is looking at the total blend of the volatile organic compounds from these different. So it's very clear that indeed, through the process of breeding, we are also changing plant chemistry. And we also saw this clustering by the heterotic group, very clear. And furthermore, too, we also were able to also see the differences grouping by the breeding era. So clearly, as we continue to do through the process of domestication, we are altering plant chemistry. And so we want to know, our next step is, then how does this influence insect-plant interactions? So we're very excited. This is also another exciting project that I am working on. But however, remember, we were trying to also see the influence of the microbe and see if we also see the strong differences that are as a result of the interactions of plant with the PGPR. We did not detect such a strong clustering based on our microbe treatment. So it is possible. We think that the genotype, the plant genotype effects, may have overwhelmed the microbiome treatment effect. So this is we're stepping back and asking ourselves, wow. I mean, we saw such strong effects of the genotype, but we're not seeing such a strong effect of the microbiome. So what does that mean? So does it mean as we continue to breed for new varieties, then we are losing the ability of plants to communicate, to have signals to the beneficial soil microbes and attract the beneficial soil microbes? And so if we are truly pushing on for the usage of beneficial microbes, yet the plants are not communicating with the microbes, then there's a disconnect. So it's important to further incorporate these factors as in the breeding pipeline, and also look at what the outcomes are. So we're fascinated. And so this is just continuing. I remember I said we dissect the blend. We want to understand both the total blend as well as individual compounds within the blend. So indeed, we see differences in the total blend, which clusters very well by the plant genotype. And as I said, we saw differences when we look at now individual compounds and break them by the genotype, by the heterotic group, by the breeding era. We see also differences. For instance, we saw like a compound methyl salicylate, which is a strong compound that is very important for attracting natural enemies. We did not detect it in some of the genotypes. So this started got our insect antennas like, wow, what's going on? So it's clearly that it's important that we look at what these alterations via breeding, how they are impacting chemistry and the subsequent plant insect interactions. So we also were able to, using Illumina, we're able to look at the abundance of bacillus groups in the different treatments. And yes, indeed, we see a colonization. However, we also saw a very strong effect only in the sterile soils, where we inoculated the plant, inoculated with PGPR on sterile soils. So it's also clear that there's a lot of competition between the native microbiome with just once we introduce our bacillus, there's a lot of competition, and that can also further affect how eventually the outcomes of this microplant insect interactions are. So it was good to know. So it's good to know that, yeah, that indeed there are many factors that are shaping this interaction. So in summary for this project, yeah, indeed, we saw that maize plant genotype does impact volatile composition, while we did not see a strong effect when it comes to the microbiome. And we see a lot of strong effects only in the sterile. So it's clear that the rhizosphere microbiome is, or may have excluded the establishment of our inoculated bacillus. And so this really lays the need for more research to understand the ecological interactions between the bacillus PGPR strains and other microbe products that are out there in the root microbiome, so that we can optimize the retention and as well as improve the efficacy of these inoculants. So we will continue to investigate this. It got really, for me, it got very exciting to see these differences that are tied to crop breeding. And so, you know, as a scientist, when you get excited, you keep going. And so from 6, we actually doubled the number. And this summer, we just finished a study with 24 genotypes that were selected to cover the major events in maize domestication history. And we're still processing the results. Yeah, so we're excited to see what our results tell us, and then we'll continue moving to further understand, you know, the interactions between the plant genotype, microbes, and how that cascades to change chemistry, and further then also how does that impact oviposition preference, growth, and performance. So it's just, these experiments are just setting me up for many more work, but much work, but we're very excited. So I think, I mean, if I've not convinced you that, yeah, indeed, it's truly a volatile, volatile world out there. And I would like, you know, as you go through your day every day, stop for a moment and think of the volatiles and plant chemistry and the conversations that are happening around you, especially as it relates to plants, insects, and all the interactions. So why am I interested in understanding plant chemistry? For me, it's when we think about food insecurity is on the rise, and before we would think about food insecurity as something that would affect different continents but not the United States, but lately it's very clear that and of course we can say we can tie it all onto COVID-19, but we know that there is increasingly pest-related challenges are on the rise, and as well as abiotic stresses such as drought and flooding that could further exacerbate the climate-induced, changing climate-induced pest-related challenges that further tie on into food insecurity. And so as a scientist that's working on plants and insects and looking at the chemistry, which is the language of communication between these two interacting, and also the beneficial soil microbes. So it's food security and our ability as humankind to feed ourselves banks on the scientific advances to deliver solutions. And so some of the applications of the work that I do can be biological control, ensuring that we do not push a lot of pesticides to control the insect pests. As I said, if we're able to genetically engineer the crops and breed crops that interact very well with beneficial soil microbes so that they change the plant chemistry, and to recruit a lot of natural enemies that can then be able to reduce the insect pest depredation. So that for me is some of the applications that can happen from and do actually happen from my research. And further, we can use chemistry to build smart sensors that are able to detect the pest as well as detect stress, and be able to then, if for example water stress, we know that water stress produces different chemical signature. And so if we can bank on such intelligence that is coming from the plants and chemistry related intelligence to be able to help plants, so that, and as well in turn, as we help plants, we are able to help ourselves as humanity and our food systems and our food supply. And of course, the technology with PPR or mycorrhizal fungi, seed coating them with strains that can change plant chemistry, make the plants grow better. So all tapping on all the benefits that come along with associations of plants with their microbe partners. So I always say, and as a person that grew up in Kenya, most of the challenges that are pest related are very personal, and I grew up seeing drought, seeing insects in action, and so I'm very dedicated and passionate about the science that I do, and it keeps me questioning. And so, yeah, with that, I hope I made you appreciate chemical ecology, volatiles, and the interactions that are mediated by these chemical signals, and from below the ground to above ground. I would like to say thank you once again for inviting me, and I would invite questions.
All right, thank you so much. We are a little bit over time, but I think we still ask a couple of questions. If anyone has questions, feel free to raise their hand in the chat here, and or put something in the question A box. Yeah. Well, if there isn't any questions, I guess I'll go ahead and say thank you so much for coming and presenting us with Scratch that. Okay. Shalonda put in the chat here, have you all considered how plants, volatiles, will change with climate change?
So that's a good question, and I didn't show that. Otherwise, I would also it would go probably past 1 hour. But yeah, indeed, actually, climate change will definitely change plant chemistry, and with that also comes also changing the plant and insect interactions that are associated with plant chemistry. So with increasing temperature, we know that plants produce more volatiles, and we know who are they inviting. Are they inviting more pests, or are they inviting natural enemies? So that will also, so unless we find out, but most of the available research is showing that with increased temperatures related to climate change, we see an increase of the herbivores. So it's inviting more of the insect pests rather than the natural enemies, and so that leads to more crop damage. And of course, also with changing climate, we know that it's also changing the temperatures and making it difficult for beneficial soil microbes to be able to also do the job that they have evolved to do. So it's gonna affect just plant chemistry on so many levels. Also change the signaling, the plant signals by which the plants produce to invite the microbes. So there's we expect that that will change, and that will in turn impact plant and insect interactions that are mediated by chemistry. And more so with also climate change, we'll also have many more interacting factors. So we know that we are preparing for a world where the crops are facing both biotic, which is a carnivory, as well as a biotic stress. So a combination of multiple stress factors, and all this is going to definitely weaken the plant's defense and the ability to defend, including the plant chemistry. And so we also may have, on the other hand, plants that cannot also produce the chemistry, the chemical signals that are so also important. And because there's also, the plant has to invest in dealing with the biotic stressors, which it has to prioritize the levels of which, the levels of defenses with which it's going to mount. So chemistry can come to the to like, okay, you to be in the back burner. So we expect so many changes that will come along with the changing climate.
That was a great question. And any other questions? I'll find more. Another question, anonymous question. Have you been able to identify, or are you interested in identifying any allies or SNPs that are associated with certain VOCs?
Definitely. We, it's just as much information as we can get about just this chemical compounds and that are produced by plants. The more holistic we get an understanding of what's going on, and so I think we continue to question and look at just different ways to get information about this volatile organic compounds. And and definitely, and the more we we involve other questions, the more we also learn about the functions and the biological functions that they play too. And probably there are many other biological and ecological functions that we haven't found out because of we're still limited in the approaches we are using to ask these questions.
Okay. Last call for questions. Okay. Well, if there's no further questions, thank you all for joining us. And thank you, Dr. Ngumbi, for coming. I will see you shortly with the graduate meeting right after this. Yeah.
Thank you so much. Thank you, and see you shortly.
Yes.